The New York Times recently wrote that the oldest human genomes reveal how a small group burst out of Africa. The article states that their skin was most likely dark, and they lacked some key mutations found in living Europeans. They did not have the genes that produce pale skin, for example, which suggests that they had dark pigmentation, as their ancestors who emerged from Africa did. A press release by the Max Planck Society for the Advancement of Science regarding the same study as the New York Times article stated that when analyzing genetic variants related to phenotypic traits, scientists found that ancient European individuals from Germany, Czechia, and Romania carried variants associated with dark skin and hair color, as well as brown eyes, reflecting the recent African origin of this early European population. The press release and the New York Times article were accompanied by images, which depict this out-of-Africa group in the frigid wastelands of Ice Age Germany. This group apparently walked all the way from Africa, barefoot as depicted in the image from the Max Planck Society. So maybe we should take their press release with a grain of salt. Did modern humans really burst out of Africa, as the New York Times wrote? Not quite. The term burst implies a sudden explosive migration. Humans didn't burst out of Africa in one sudden surge. But what about the study itself? Is there good science when we dig deep into the study? The answer is yes, but it contradicts with the images from the press release. Imagine a small band of early humans, fewer than 1,000 people, walking out of Africa around 130,000 years ago, venturing into unfamiliar lands. This isn't the big migration most people think of, which happened around 80,000 years ago, but a smaller, earlier burst from northeastern Africa, the Horn of Africa or North Africa's coast, into Arabia. Scientists call this the Out of Africa event, and this particular moment, suggested by some studies such as the Arabian standstill hypothesis, happened during a warm, wet period about 130,000 years ago. Back then, the Sahara was greener, with lakes and grasslands, making it easier for these folks to cross into what's now Arabia. This small group didn't have the numbers or tools of later humans, but they were curious and brave, taking their first steps into a new world. However, this early migration 130,000 years ago might not have lasted. Some geneticists think they either died out or got mixed into later migrations because we don't see their DNA much in people today. But in 2016, a study found evidence of their genes in Australian Aboriginals, around 2% of their genes, in fact. When that study was released, geneticists Joshua Akey and David Reich of Harvard dismissed the study because it contradicted with their simplistic idea of a single migration. Akey even stated, "As geneticists we can spend the next decade arguing about that 2%, but in practical terms it doesn't matter." This is a really incredible statement from a geneticist. 2% doesn't matter. That is the same amount of DNA we have from Neanderthals, and he is saying it doesn't matter. Ridiculous. Here we are almost 10 years later, and we are still talking about that 2%, so it does really matter. Indeed, this 2% is a fascinating peek into how humans started spreading across the globe, driven by changing climates and a need to explore. This group wasn't huge, maybe just a few families or tribes, and they carried with them the basic traits and genes of their ancestors. Their skin was likely a medium to dark brown, not as dark as some modern sub-Saharan Africans, because northern Africa, where they came from, gets less intense sunlight than deeper tropical areas. They used simple stone tools, like the Levallois method, and relied on fire and shelters to survive in the deserts and grasslands they encountered. But life was tough. Climate shifts after their departure, like cooling and drying around 115,000 years ago, would have made it hard for them to stick around, leading to their disappearance or blending into other groups much later. This small burst out of Africa sets the stage for understanding who these early humans were, how they were related, and what they looked like thousands of years later. Now let's talk about the DNA of these early humans, focusing on something called haplogroups, which are like family trees for our genes. Haplogroups are groups of people who share a common ancestor because they inherited the same unique changes or mutations in their DNA. For the small group that burst out of Africa around 130,000 years ago, their haplogroups would trace back to their roots, mostly from mitochondrial DNA, passed from mothers, and Y chromosomes, passed from fathers. As they spread out, their close relationships and small population size became really important. This group was tiny, so everyone was closely related, like cousins or even siblings in some cases. Their DNA shows this because there wasn't much variety. It was like a small family sticking together. Scientists can tell from genetic studies that small populations, like this one, have less genetic diversity because there aren't many people to mix genes with. This close-knit group would have shared similar traits, like pigmentation and height, because they all came from the same African ancestors and hadn't had much chance to mix with others yet. Around 80,000 years ago, they weren't very tall compared to us today. Scientists estimate that early modern human males stood about only 5'3" on average. This shorter stature makes sense because these humans lived in the subtropics of southern Arabia, where their bodies evolved for survival in hot, tropical environments, but not necessarily for being super tall. Their height was shaped by genetics, diet, and lifestyle. Think smaller builds for agility in open savannas of the Green Arabia and Sahara, eating a mix of plants and small game, without the protein-rich diets or cold-weather adaptations of later humans. This contrasts with their taller and more robust descendants in Eurasia, such as the Cro-Magnons. The small size also made them vulnerable. With so few people, they faced risks like inbreeding, where close relatives have babies, which can cause health problems or environmental challenges like harsh climates or food shortages that could wipe them out. Genetic studies like those in the Bachokiro and Oasis studies show how later groups around 45,000 years ago had more diversity because they were larger and interbreed with Neanderthals. But for this 130,000-year group, their tight-knit small population meant their DNA was like a snapshot of early human genetics at the time. Simple, shared, and shaped by their origins, but limited by their numbers. The press release mentions that they likely had dark pigmentation. However, dark is a broad and imprecise term that can encompass a range of phenotypes, from Mediterranean or Near Eastern tones to deeper sub-Saharan African shades. Skin pigmentation is polygenic, controlled by multiple genes with additive and interactive effects, rather than a single locus. The absence of specific light skin alleles like SLC24A5 or SLC45A2 doesn't automatically imply dark skin. It simply means the individual lacks those particular European-derived lightening variants, leaving the phenotype dependent on other genetic and environmental factors. Assuming that the first modern humans had skin as dark as the midnight sun is fundamentally flawed and oversimplifies the complex evolution of human skin pigmentation. This assumption ignores the polygenic adaptive nature of skin color, which responds to environmental pressures like UV radiation, and overlooks the historical and genetic diversity across human groups. Instead, evidence suggests that the earliest modern human populations likely had a range of skin tones, and that darker skin variants evolved more recently in some African populations, including the Yoruba of tropical West Africa, as adaptations to specific ecological niches. So the question remains, what did the basal or ancestral out-of-Africa population look like? Would they fit into a Nubian fresco, or have a more intermediate skin tone such as the Bushmen of Central Africa? In fact, genetic studies indicate that some dark skin variants emerged relatively recently in human evolution from lighter-skinned ancestors. For instance, the derived alleles at SLC24A5 and SLC45A2, which lightened skin, emerged 50,000 to 60,000 years ago, but their ancestral dark forms are still prevalent in Africa. However, darker skin alleles, like those enhancing MC1R or TYR activity for high melanin production, may have evolved more recently in sub-Saharan Africa as adaptations to intense equatorial UV, particularly after population divergence. If the Yoruba of West Africa, estimated to have diverged from other African groups around 130,000 years ago, as suggested in some genetic studies, originally had lighter skin, their darker skin today would reflect subsequent selection for increased melanin to protect against UV damage in West Africa's equatorial climate. This population diverged from those outside of Africa around 100,000 years ago, and genetic studies confirm that they are not ancestral to non-Africans. They are a sister lineage. This evolutionary trajectory is supported by the example of the Bushman and Pygmy populations in southern and central Africa, who, despite living in high UV regions, exhibit lighter skin compared to West African groups like the Yoruba. This lighter pigmentation in Bushmen and Pygmies may result from adaptations to lower UV exposure in forested or arid environments, or genetic drift, suggesting that dark skin isn't universal even within Africa. Similarly, North African populations, historically more moderately pigmented, indicate that early humans might not have started as dark, but as a lighter shade before darker variants evolved in some regions. As these groups moved into Europe and Asia, Neanderthal admixture introduced alleles like MC1R that could lighten skin slightly, but without the derived variants, some remained darker than later Eurasians. In short, assuming the first out-of-Africa crew was as dark as the midnight sun is wrong, it's more likely they had moderately pigmented skin like modern Northern Africans or some Bushman Pygmies. Northern Africa's history of moderate pigmentation and diverse African populations like the Bushman Pygmies show that dark skin isn't a universal African trait, but an evolved response to specific environments. Thinking the first out-of-Africa travelers were midnight sundark is like assuming all Africans today look exactly the same. But with Northern Africa's olive tones and the Bushman's lighter browns, they were probably not as dark as depicted by the New York Times. But why does this even matter? Because science is about facts. Fast forward to about 45,000 years ago, and we find ancient humans whose DNA tells us a lot about skin color. Oase I from Romania lived long after that early 130,000 year out of Africa burst, but his genes still carry clues about his roots and mixing with Neanderthals, which changed his skin tones. Let's break it down in simple terms, using a scale called the Monk's skin color chart. Pestera su Oase means the cave with bones in Romanian, so the fossil known as Oase I literally means the bone. Oase I, who lived around 41,860 to 36,950 years ago in Romania, had a whopping 6.9% Neanderthal DNA, mixed in just about 100 to 150 years before he was born. Therefore, Oase I was already pure European. Starting with the dark brown skin of their ancestors that rarely burns and tans very easily, their Neanderthal genes gave their skin a big lightening boost. These Neanderthal genes came in long stretches, covering big chunks of their DNA like 1 to 10 million base pairs, on chromosomes with genes that control skin color, especially genes on chromosomes 9, 11, 12, 15, and 16. If Oase I had the usual single copy of these Neanderthal genes, called heterozygous, their skin would lose about 40 to 60 melanin units, dropping from 80 units 20 to 40 units, meaning light brown skin that usually burns and tans with difficulty or gradually. If, by a rare chance, less than 1%, they had 2 copies of the Neanderthal MC1R gene, their skin could become really pale, but that's super unlikely. With the low sunlight in Ice Age Romania, about 45 degrees north, skin might have gotten a little lighter by losing 5 melanin units, but summer tanning could add back 10 units, keeping in the middle range. So, Oase I probably had pale to light brown skin, much lighter than their dark-skinned ancestors, thanks to that big Neanderthal genetic punch. Hair color is also polygenic, controlled by many of the same genes which influence skin pigmentation. Neanderthals are known to carry variants that influence lighter or reddish hair, and Oase I's Neanderthal DNA also impacts this trait. So Oase I likely had medium to light brown hair with possible reddish undertones, such as auburn or chestnut, due to heterozygous Neanderthal variants. This aligns with a lighter phenotype, but not blonde or red like some modern Europeans. Hair curl is influenced by several genes, with curliness linked to African ancestry and straightness to Neanderthal ancestry. The chromosome chart shows blue segments on chromosome 1, suggesting a Neanderthal variant. This could result in wavy to straight hair, and blue segments on chromosome 2 suggest a Neanderthal allele, further reducing curliness. Furthermore, Oase I likely had dark brown to hazel eyes due to Neanderthal OCA2 and TYR variants, but not blue or green, reflecting brown-eyed ancestry. Indeed, Oase I's dark brown to hazel peepers, tweaked by his Neanderthal DNA, might have caught a Neanderthal maiden's eye. This genome also has long and frequent Neanderthal DNA segments on chromosomes 1, 2, 10, and 15. These long strands are highly likely to contain or influence Neanderthal introgressed variants that directly or indirectly affect testosterone production or regulation. Testosterone influences a variety of male traits, including height, aggressiveness, bone density, muscularity, and strength. The genome also contains Neanderthal introgressed tracts in the region of chromosome 1 associated with blood type. Of the 3 Neanderthals tested so far, all 3 have a unique blood type known as rhesus incomplete, which is only found in some Australian and Papuan Aboriginals. So, our guy may have had Neanderthal blood type coursing through his icy veins. Bang, just when you thought genetics was boring. Height is also highly polygenic, with Neanderthals generally taller than early modern humans. This might surprise you, because it is commonly stated that Neanderthals are short, but Neanderthals had height-increasing alleles on chromosomes 12 and 6, potentially adding 1 to 3 inches. OSA1's height, stretched by his Neanderthal DNA, might have towered over his peers, but he was still short of a Neanderthal giant. Lip shape is also influenced by several genes, with African ancestry often linked to fuller lips and Neanderthals to thinner lips. Based on his genetic profile, OASE1 likely had moderately full lips, less pronounced than his ancestors' fuller lips, but not as thin as a Neanderthal's. This fits a hybrid Ice Age European face, blending ancestral and Neanderthal traits. So OASE1 likely had straight or slightly wavy light brown hair, olive to light brown skin, lighter than his ancestors but not as pale as modern Northern Europeans, thanks to a moderate Neanderthal genetic tweak. Think a prehistoric Mediterranean tan, dodging burns but soaking up the rare sun. This is an example of how individual evolution recalls and replicates the evolution of the ancestral race. OASE1 represents a stage in this evolution that is unquestionably distant from the beginning, resembling the modern state of man while remaining distinct from it. OASE1 shows how Neanderthal DNA, mixed in recently, lightened their skin from a dark brown start, but the exact shades depended on how much Neanderthal DNA he had and the sunlight he got. Neanderthal introgression in modern humans typically spans all chromosomes except the Y chromosome, due to male infertility issues in hybrids. Neanderthal Y DNA is absent in modern humans due to male hybrid infertility or strong selection against it. In mammal hybrids, think about hybrid horse-donkey mules, male offspring are often sterile due to genetic incompatibilities. Neanderthal modern human hybrids likely face similar issues, particularly males carrying Neanderthal Y chromosomes. The Neanderthal Y chromosome differs from modern human Y at several key genes. Studies suggest a Neanderthal Y chromosome had mutations incompatible with modern human autosomal genes, causing sterility or immune rejection in male hybrids. Fertile female hybrids could pass on autosomal Neanderthal DNA, but male hybrids couldn't reproduce, purging Neanderthal Y from the gene pool. Therefore, if Neanderthal male hybrids were infertile, modern human women would have ditched Neanderthal dates faster than a bad Tinder date. Whatever the case, OASE1 belongs to an ancient Y-DNA haplogroup and an extinct mitochondrial DNA haplogroup. OASE1's paternal Y-DNA belongs to haplogroup K2A. Haplogroup K2A is a major branch of haplogroup K. Haplogroup K is thought to have originated in Southwest Asia or Central Asia around 47,000 to 50,000 years ago, with its descendants spreading widely across Eurasia. OASE1's maternal lineage is related to mitochondrial DNA haplogroup N, but diverged from all other N clades before they diverged from each other. Now let's circle back to that early 130,000 year out of Africa burst and what it means through the lens of the Arabian standstill hypothesis. This idea suggests that after leaving Africa around 130,000 years ago, that small group hung out in Arabia for a while, maybe 50,000 to 80,000 years before moving on. This hypothesis ties into the warm, wet climate of that time, and it has big implications for understanding human evolution. If humans burst out of Africa 130,000 years ago, it means they took advantage of a unique time called the Eemian Interglacial, around 130,000 to 115,000 years ago. Back then, the Sahara and Arabian Peninsula were lush, with lakes, grasslands, and fewer deserts, thanks to warmer global temperatures and more rain. This Arabian humid period made it easier for a small group, fewer than 1,000 people, to cross into southern Arabia from northeastern Africa. The Arabian standstill hypothesis says they might have stayed in Arabia for a long time, adapting to that environment before continuing their journey. This fits with burials and tools found at places like Skul and Kafse, dating to 120,000 to 90,000 years ago, showing early modern humans with simple stone tools, like the Levallois method, living there during this warm period. But here's the catch. This early group probably didn't make it big. As we discussed earlier, their small size, maybe just a few families, meant they were closely related, with little genetic variety, making them vulnerable to climate changes or competition. After the Eemian, around 115,000 years ago, the climate cooled and dried out, turning Arabia back into a desert. That could have trapped these early humans, or they might have gotten absorbed into later migrations around 80,000 years ago, when a bigger group left Africa during another warm period. Genetic studies don't show much of their DNA today, suggesting they either went extinct or left only faint traces, like in some ancient bones, but not in modern Eurasian genes, except in Aboriginal Australians and Papuans. For pigmentation, this 130,000-year event is key. These early humans likely had medium to dark brown skin because they came from northern Africa, where sunlight isn't as intense as in central or southern Africa. They didn't need skin very dark, which evolved later in some African groups under stronger UV. In Arabia's moderate sunlight, their skin might have stayed the same or gotten a little lighter, but without mixing with Neanderthals yet, since Neanderthals were mostly in Europe and Central Asia, their pigmentation wouldn't have changed much. If they stuck around in Arabia, the lower UV there would have pushed their skin toward lighter shades over tens of thousands of years. During the Ice Age in Europe, which spanned roughly from 115,000 to 11,700 years ago, environmental conditions were harsh, with long winters, limited sunlight, and frigid temperatures dominating much of the region. In the high-latitude environments of Ice Age Europe, where sunlight was scarce for much of the year, darker skin, which contains more melanin and blocks ultraviolet radiation, would have hindered the body's ability to produce sufficient vitamin D from the limited sunlight available. Lighter skin, with less melanin, allowed for greater UV penetration and thus more efficient vitamin D production, giving a survival advantage to those who carried this trait. For infants, this adaptation was especially crucial. Vitamin D deficiency can lead to a condition that weakens bones, stunts growth and infections, outcomes that would have been deadly in the unforgiving Ice Age environment. Infants rely heavily on their mothers for vitamin D, either through breast milk or indirectly through the mother's own stores built up via sunlight exposure. In populations with darker skin migrating from sunnier regions, the reduced UV exposure in Europe would have resulted in insufficient vitamin D levels, jeopardizing infant survival. Natural selection favored lighter-skinned individuals whose offspring had stronger bones and better overall health, allowing them to endure the cold, resource-scarce conditions. Over generations, this pressure helped lighter skin become a prevalent trait among European populations, a legacy still visible today. These new studies are consistent with the paleontologically established presence of the last common ancestor of Neanderthals and modern humans in Eurasia, a Eurasian divergence between Homo sapiens and Neanderthals 800,000 years ago, a mitochondrial DNA introgression from Homo sapiens into Neanderthals 500,000 years ago, and a Eurasian divergence among the ancestors of living humans 250,000 years, and the exodus of the Bushmen into Africa, and the presence of modern humans in Arabia and Persia 130,000 years ago. The molecular out-of-Africa hypothesis has been considered as an established fact among population geneticists for some 35 years, despite concern with it among phylogeneticists with experience beyond that of the human genus. The paleontological support for the hypothesis is also questionable, a circumstance that in the light of expanding Eurasian paleontological knowledge has become accentuated through the last decades. So, our species is much older and more diverse than we once believed, and that is very exciting. Let us know what you think in the comments, and please share and subscribe.